clinical genetics. So before discussion of clinical genetics, we should know the gene. Gene means sequence of DNA or polymer of nucleotide is called as gene. Gene is single gene containing the 800 to 900 base pairs. So in base pairs, codons are present. One codon is equal to triplet. Triplet means 3 bases or amino acids. Example ATG. So this type of codons arranged in sequential manner, that is known as gene. So next, genetics. Genetics means study of DNA or study of gene or sequence of DNA, so which are arranged in sequential manner, that is the genetics. From the genetic we can identify the sequence of DNA and characterize the DNA and identify the number of chromosomes in various organisms. So in genetic, especially genetic material is nothing but DNA. So DNA means deoxyribonucleic acid. DNA means deoxyribonucleic acid. So DNA is the DNA present in center of cell, which is present in nucleus. In nucleus, two types of genetic materials are, which are DNA and RNA. So DNA containing the various components like adenine, guanine, cytosine and uracil. So these materials are present in DNA. In case of RNA, uracil is there. The difference between DNA and RNA, the DNA containing the adenine, guanine, cytosine, uracil. But in case of RNA, only uracil is present along with these base pair also present. So in between DNA and RNA, some variance is there except uracil. So these are the components of DNA. So DNA containing the various materials like nucleotides, nucleosides, sugars and phosphates. So these materials are skeleton of DNA. So every nucleotide, nucleoside, sugars, phosphates attached with hydrogen bond. According to charge of rule, purine basis is equal to, in every organism, purine basis is equal to pyrimidine basis. Purine basis equal to pyrimidine basis. So this is the charge of rule. So this is applicable for the every organism in every species. This is the universal rule for every organism. But in human being, the chromosomes are 46. In human being, chromosome number is 46. This chromosome number in human being, 46 chromosome numbers are there. But in case of birds, animals, plants, it varies different species of organism. So chromosome means bundle of DNA fiber. Chromosome means bundle of DNA fiber. Chromosome means DNA Chromosome means DNA bundle is called as chromosome. This chromosome number varies with species to species. In human being, DNA, the chromosome number is 46. In case of birds, you can take pigeon. This pigeon containing the highest chromosome in entire world. Pigeon containing the 80 chromosome number. Whereas in case of plants, you have to take tobacco. Tobacco containing the 48 chromosome number. But in case of genetics and clinical genetics, so difference between clinical genetics and genetics, somewhat difference is there. In genetics, we can study and identify the characteristics of DNA and observing the sequence of DNA and identify the particular sequence of DNA. If any defect of DNA occur in the chromosome, it produces the defect of DNA. So in clinical genetics, we can study the sequence of DNA and observing the lack of DNA segments. Due to the lack of DNA produce some genetic diseases like cancer, diabetes, rectal cancer, intestinal cancer, nerve tubule disease, so these are the genetic defect diseases. So how it will be occurred? Due to the lack of some traits. These traits, when absent in DNA material, it produces the defect of DNA disease like hereditary disease. These hereditary diseases are like cancer, diabetes, rectal cancer, intestinal cancer and nerve tubule disease. So these the hereditary material, when it is lack of DNA occur in particular segment of DNA, produce the defect of DNA causes genetic disease. These genetic information, when it is lacked in cell, automatically then that information passes from one generation to another generation. So from this study, we can identify the different types of disease like life-threatening disease. In life-threatening disease, especially cancer is the life-threatening disease. In entire world, so many people are facing cancer, diabetes, rectal cancer, intestinal cancer, nerve tubule disease. Like defect of DNA produces various life-threatening diseases like cancer, diabetes, intestinal cancer, rectal cancer, nerve tubule cancer, etc. These entire diseases spread over the world due to the lack of traits in DNA segment. For example, DNA is the linear in shape. It contains the set of codons. When this segment losses, due to the loss of this segment produces the false DNA produced. So due to the false DNA causes defect of DNA. So this DNA causes lack of disease, and this lack of disease giving the wrong information to cell. That cell signaling passes the wrong information to entire cell. That information, wrong generation passes in cell. The signal leads to one cell to other cell. Likewise, at the same time, the DNA defect producing the various disease, that disease passes from one generation to another generation. Like if any parent containing the diabetes, that symptoms also passes to children. Likewise, all disease, defective DNA defect producing disease passes from parent to children. So this is the clinical genetics. Next, from the clinical genetics, we can identify the various diagnosis tests. Like some tests are particularly used for the defect of DNA producing disease. For example, some tests are like pre-neonatal test, and forensic test. These forensic tests can be used to identify the life-threatening disease like cancer, diabetes, rectal cancer, and biopsy tests. So all these tests are confirmed to identify the DNA defecting diseases. Next, DNA replication. In DNA replication, DNA containing the certain fixed point started at origin. So this fixed point is known as origin. At origin, certain enzymes are binded at origin point. DNA helicase, DNA gyrase. These two enzymes melted the origin at DNA. This melting DNA produces two Y-shaped forks of DNA are produced. So this reason is the Y shape of DNA fork. This is the second DNA template containing the Y-shaped DNA fork. So this, when melting occurs with these two enzymes, it produces the two Y-shaped DNA forks. In second step, some proteins are there. SSBP means single stranded binding protein. This single stranded binding protein maintain the metabolism of DNA or stabilize the DNA material. In next step, this DNA again bind with primase enzyme. This DNA again bind with primase enzyme. This primase enzyme initiate the RNA primer. Again the RNA primer synthesize the 3' hydroxyl group. This 3' hydroxyl group increase the addition of nucleotide of DNA. The nucleotide of DNA produce the new DNA. It produces the new DNA. So this strand is called as leading strand. Simultaneously, when DNA is progressed, that strand is called as leading strand. Again, simultaneously, primase enzyme binding at lagging strand. The second strand of DNA is called as lagging strand. This strand is called as lagging strand. So the primase enzyme again binding on second lagging strand. This lagging strand again initiate the RNA primer. The RNA primer again synthesize the 3' hydroxyl group. This 3' hydroxyl group produce the small fragments of DNA. The small fragments of DNA is called as Okazaki fragment. The small fragments of DNA is called as Okazaki fragments. These Okazaki fragments are small DNA fragments. Okazaki fragments are called as Okazaki fragments. These Okazaki fragments again combined with DNA polymerase enzyme. DNA enzyme 1. This Okazaki fragment digests the DNA polymerase enzyme to produce the fragment of new DNA. this new DNA generated from the lagging strand. At the same time, this new DNA occur in every organism. So this entire phenomena, this entire mechanism occur in either prokaryotic cell or eukaryotic cell. So this is the DNA replication. This is the general model of DNA replication for entire mechanism. Stop it.